We're a team of 5 undergrads uncovering research here at Dundee University.
A lot of important research goes on in the building behind us and in various other places throughout campus. More specifically, we're going to be looking at Dr Stavridis' work into retinoic acid's influence in fibroblast growth factors, signalling and its influence on stem cell differentiation. What we found is that it's a field of almost unlimited potential, and this is partially due to what embryonic stem cells are.
Stem cells are cells that have a choice, okay? And they have a choice in what they're going to do. They can stay as stem cells or they can change and become something different.
As Dr Stavridis said, these cells have a choice.
Choosing between becoming fat, liver, muscle, white blood cells, red blood cells, skin, neurons, or glial. There are many pathways of cell signaling. This research focuses on the FGF pathway in relation to retinoic acid's effect on embryonic stem cell differentiation.
However, retinoic acid is special, as it can bring about cell differentiation through non-genomic methods. For instance, gene transcription is not required for retinoic acid to have an effect. Also, retinoic acid has the ability to induce cell differentiation after the cell has lost the ability to self-renew.
There is evidence to suggest that retinoic acid represses the pluripotency gene OCT4. OCT4 and SOX2, a transcription factor required to continue embryonic stem cell self-renewal, work in conjunction to promote FGF4 expression in embryonic stem cells. In the embryonic axis, retinoic acid downregulates FGF4 expression. Retinoic acid and FGF4 signaling are mutually inhibitory.
Retinoic acid receptors are nuclear receptors, which may also act as transcription factors. These retinoic acid receptors are often involved in the way that retinoic acid induces a cellular change, although they are not always necessary, as the retinoic acid can induce a change non-genomically. This method is how ERK signaling is activated.
A signaling pathway functions with 4 components. The first one is a ligand. It is a signaling molecule which will bind to the receptor. The receptor is a molecule that receives the signal. Then the transducer appears. It will link the receptor to the effector, and the effector is a molecule that causes the cell to change.
These 4 components make up the basis of all cell signaling pathways.
Each of these 4 components has been identified and are displayed here in this diagram.
The ligand is called FGF and binds to the FGF receptor or the FGFR. This binding activates the transducer, ERK, AKT, to communicate and signal the effector. The effector, which is ETS, will then transcribe genes. Signaling from this pathway activates transcription of genes that promotes differentiation.
Understanding the basic components of the FGF pathway is essential for understanding how other factors, such as retinoic acid, affect and influence cell differentiation.
Prior to this research, the mechanism of action of retinoic acid in relation to FGF and ERK signaling was poorly understood. Now, the research has given some insight into the extent to which retinoic acid is involved in the timely acquisition of somatic cell fate in embryonic stem cells.
Additionally, retinoic acid was shown to promote differentiation by a dual mechanism. Primarily, retinoic acid increases FGF signaling. Then, approximately 24 hours after FGF signaling is blocked, differentiation continues despite ongoing ERK signaling.
This graph depicts the gene expression profile of key genes during embryonic stem cell differentiation. As shown, retinoic acid stimulates the upregulation of FGF5 and FGF8, while FGF4 is downregulated.
So I'm going to explain the main discovery that the author found. They found that retinoic acid promotes cell differentiation by opposite action. Indeed, ERA initially stimulates FGF signaling in cell in day 1. And we can see that in figure 2.a, and that signal leads cells to differentiate. However, after a period of endogenous FGF activity, retinoic acid attenuates this signal. We can see that on figure 3.c, and that signal leads also cells to differentiate. So this discovery was surprising, and the author explained that by the fact that ES cells on day 1 and on day 2 are different, and that's why the response to retinoic acid is also different. Now the authors have a better understanding of the mechanism that leads cells to differentiate, and that knowledge could be used for screening drugs, modeling disease, and for new cell therapy. However, the impact of the article is limited for several reasons. First, this research is only the first step in the comprehension of the complex process of cell differentiation. Besides, the author during the research have worked on mouse ES cells and not human cells, and this model is not ready to be used for transplantation on humans.
We would like to give a special thank you to Dr Marios Stavridis.
Dr. David Booth.
And Dr. David Martin.
Thank
Thank
you.
you.